I wanted to talk to you today a little bit about the dark side of carrier screening. Again, my name is Brad, Director of Research at Asuragen. So about 20 years ago, when my wife was pregnant with our first daughter, the new CFTR test, genetic screening test, became available. And she was tested for 23 targeted variants. We were lucky. She was not a carrier for those 23. About a year ago, a friend of mine also did a carrier screening study. She got tested for 174 variants. She was a carrier. Her partner was not a carrier for the exact same disorder. So luckily, they had low risk. A lot of families do not get that opportunity. And the guidelines are constantly changing to incorporate variants and genes that are important for carrier screening. And today I'm going to talk to you a little bit about a kit that we're developing to help shed some light on some of the harder to decipher genes and also incorporate some of the easy to decipher genes. And I argue that we shouldn't leave carrier screening to chance. So thank you, Dan, for helping me present this first slide. I don't have to really talk to you about carrier screening and talking about autosomal recessive disorders. Everyone in this room probably is a carrier for a disease. There are over 2,000 genetic disorders that have been described with Mendelian inheritance, autosomal recessive inheritance, and the prevalence of certain genes differ by different populations. And the ACMG currently recommends a 113 gene panel for this carrier screening. So typically a mother who's expecting or planning a family will go into an office and get blood drawn. That blood will go to a lab, and that lab will then test that blood for the variants of interest. In that panel, whatever panel's tested, 176 genes, 10 genes, 3 genes, whatever it is, the top variants, the top genes in that panel are typically challenging for next generation sequencing, for short read sequencing. They either have long repeats like Fragile X, or they have copy number variations, or they have different SNVs, they have different structural variants. And so what labs typically have to do is they have to run a number of different assays, not just NGS, but a number of different assays, including PCR-CE, qPCR, MLPA, long-range PCR Sanger sequencing, or some labs just don't offer those. Typically, this carrier screening is done in a centralized laboratory, and then the results are reported back. So when my wife did this carrier screening for CFTR, it took about a month to get the data back 20 years ago. When my friend did this, it took about 2 to 3 weeks to get that data back. And we're hopefully developing a kit sample to answer in 3 days using Nanopore sequencing. That carrier panel is basically a panel on a platform that can cover all of those different assays in one method, one platform, using Nanopore sequencing. So we have 11 genes on that panel. We use amplification, enrichment, PCR enrichment, long-range sequencing, and then we've built software that can help with that analysis. So the reporting is done automatically. We can cover a lot of different genetic variation, including SNVs and indels, more common for NGS, but copy number variation, structural variation, paralog deconvolution, exon del dupe identification, and repeat sizing, along with interrupt sizing. And so those bands around that graph on the right-hand side, those are typically done by different tests currently by testing labs. So this will help out those testing labs. We've tested that in a poster number 5 here in the conference, number 12 online. That has a bit of the data that's wrapped into this figure. We've tested on a number of whole blood and cell line samples with excellent accuracy so far in our alpha test, and we are looking to expand that out a bit more. So in this unified workflow, we're providing simpler, empowering tools for the lab. We're providing better answers for the clinician and faster results for the patient. Thank you very much.